Okay, good morning. Today we are going to do the final review for Unit 4, Cell Communication. And the cell cycle. All right, let's begin. Just in humans alone, the human body is approximately, and I mean a full-grown human body, is approximately 100 trillion cells. You have about 200, 250 different types, but all in all, you have 100 trillion cells. Let me make sure of that we got hundreds, thousands, millions, billions, trillions. Good. So these cells must be able to communicate with each other. You step on a Lego, which is like the worst pain on earth, you feel it in your brain. How does that happen? How do cells communicate with each other? You're being chased by a dog and you feel your heart beating very, 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 very fast. And you're either going to run for your life, you're going to stand your ground, or you're texting while driving, which none of you should be doing, and you come within inches of getting in a very bad car wreck, and you can feel like every cell in your body, the hair on the back of your neck is standing up, your heart is about to burst out of your chest. That's adrenaline. How is it that a hormone caused like my whole body to just wake up like that? I can feel every cell. That's done through cell communication. So cells communicate through cell-to-cell contact. Now, some contact can be short range, and this is generally just done by signaling. And then long range, which is done by hormones. Now, I want to talk just a couple minutes about hormones and how they're actually, what they are. You guys did not have to learn about the endocrine system in this class. It used to be a requirement, but it no longer is. A hormone is a signal that is released by one part of your body and it is meant to target another part of your body. So hormones are released by glands. Some glands include your pituitary, your thyroid, your pancreas, your adrenals, testes, and ovaries. Am I missing any? Your pineal gland, your parathyroid. Those are all structures that secrete hormones. And hormones have a specific target. Now that target could be a specific type of cell or tissue or even organ. For example, adrenaline is secreted by your adrenal glands. AD means on top of, and renal means kidneys. These are glands on top of your kidneys, adrenal, and they secrete adrenaline. Adrenaline is released into your bloodstream and it will target your heart in your cardiac muscle cells, and to make your muscle cells in your heart beat really, really fast. So let's say that you are being chased by a big snarling dog. You're likely going to have a lot of adrenaline pumping through your body because you had that fight or flight response. You're either going to run as fast as you can, and adrenaline is going to help fill your muscles in your legs with oxygenated blood so you can run faster than you've ever run before. Or you could stand your ground and fight, and your adrenaline is going to help your heart pump oxygenated blood into your muscles so you can stand there and for a very short while be like a super self. There's been documented cases of little moms, little mothers, lifting hundreds of pounds off their child who was trapped, like underneath a tractor or a lawnmower, and they're all that the mother hears is, "Mommy, mommy, help me." And just that adrenaline of being a mom overwhelms them. They pick up this big vehicle. That's been documented, like maybe up to several hundred pounds, this little old woman, because her little child is screaming. Well, hormones travel through the blood. You know, ladies, one day you might be a mother. And on the day that you give birth, you're going to be pushing. You know, the uterus is right here. But you're going to have a hormone in your brain called oxytocin. It's in your pituitary. It is going to travel down your bloodstream into your uterus to make your uterus actually contract. This would be considered a long distance signal. And it's going to continue to contract until the baby is out of there. That's just one of many examples of long distance signals. All right, next we're going to talk about signal transduction. Signal transduction is all about passing a signal along until it reaches its target. Let's say that Quinn he was sitting here in class and his mom calls the front office and says Quinn needs to be signed out at noon. So the secretary is going to give a TA, a teacher's assistant, a gold pass. And that gold pass is going to get marched up through main hall, go outside, upstairs into South Hall, and come to my room, 232. That is the transduction of a signal. It's just being passed on from mom to secretary, from secretary to TA, from TA to me, Thorson, from Thorson to Quinn. That is signal transduction. You're just passing a signal along until it finally gets to its destination. These signals are called ligands. A ligand is a chemical message. Now, ligands, the word ligand is an umbrella term for cell signals. Every ligand is different, and it is recognized by a receptor, which, by the way, is a protein, which I'll talk about momentarily, in a target cell. So let's say we have a cell right here, and here is a receptor, and the receptor looks like this. I have lots of different ligands. I have this guy, I have this guy, I have that kind of signal, and then I have this one. This is the one that, wrong color. This is the one that you want to use because that fits into that receptor. All these other ones, nope, 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 nope. Those won't fit. They are not meant for that one. Just like my car keys are meant for my car, not Mr. Bush's car, not Miss Hellenberg's car, not Mr. Webb's car, my car. Every key has a lock that goes with it. Every ligand has a receptor that goes with it. So let's talk about G protein coupled receptors. These are on the cell surface. A ligand binds to the receptor. This begins the signal transduction. It's like when Quinn's mom calls the school, Quinn needs to be signed out at noon. That begins the signal transduction, where it goes from secretary to TA, TA to Thorson, Thorson to Quinn. That's the transduction pathway. Now, one of the checkpoints that this ligand is going to hit are called secondary messengers. A secondary messenger passes on the signal and it relays it to where it's supposed to go. So in my analogy of Quinn being signed out, the secretary was the primary messenger. She's the first one to have received the signal. But then when she gives it to the TA, that little gold pass, He's a secondary messenger. And then the TA gives me the pass. I'm a secondary messenger until it gets to Quinn, who is the target. So anything that receives a signal after it's initially received is known as a secondary messenger. And the secondary messenger that we learn in class is called CAMP. This stands for cyclic adenosine monophosphate. You've learned about ATP. You've learned about ADP. Well, there is such thing as AMP. AMP, as you can hopefully decipher from the name, is a adenosine monophosphate. It has one phosphate. This is a secondary messenger. CAMP relays and amplifies signals. So if Quinn were here in class right now and the message is Quinn needs to go home at noon, that's the message. CAMP is going to say, Quinn needs to go home at noon. Quinn needs to go home at noon. Quinn needs to go home at noon. It just amplifies it and it relays it. It passes it on and it makes it more numerous until you get the job done. You know, in military, you might have a general tell a lieutenant who's a lower ranking officer. The general tells the lieutenant, tell all the soldiers, get in formation. And then the lieutenant just screams at him, everybody in formation right now. He's amplifying that message and he's relaying the message. If you guys have ever been part of a sporting team, football, baseball, you got the coach who might tell his assistant coaches to do something, and then the assistant coaches are the ones that start yelling at you and telling you to get in formation or to get to doing an exercise or to do something right, you're doing something wrong. You seldom see the head coach do that. That's what the assistants are for. They listen to what the head coach says, they pass that message on. All right. So what I would like to do now is I want to show you the difference between a polar message, or I should say a polar signal, excuse me, and a non-polar signal, and what these signals' intent are. Kylie, what type of cells are in your brain? Are they somatic cells or are they gametes? They are somatic cells. They are body cells. Do your body cells have all of your DNA or half of it? All of it. Now, in your brain right now, do you have a kneecap in there? No. Easy answer, no, I do not have a kneecap in my brain. Are the genes to make a kneecap present in your brain cells? Yes, the genes to make a kneecap in your leg are present in your brain cells. Here's the big question. Are those genes turned on in your brain? Could they be turned on? Yeah, you can turn them on, but we don't want to grow a kneecap in your brain. But the genes are there. And so, everybody, the purpose of these signals is to activate a gene that you have the whole time. It is there. Just turn it on or turn it off. That's what these signals do. And instead of saying we're going to turn a gene on or off, we don't say on and off. We say we're going to express it or not express it. So when it comes to a polar signal, let's talk about this first. Here is a plasma membrane. Kylie, what is the polarity of a plasma membrane? It is nonpolar. What makes it nonpolar? The fatty acids in the phospholipid bilayer. Okay. Well, let's say that I have this yellow polar signal. Can a polar signal pass through a nonpolar membrane? You all at home should be answering with a big shaking of your head, no, it cannot. So we have to have a receptor. This receptor is going to be the beginning of a cascade of messaging. So let's say that this messenger passes on a signal and it goes to a secondary messenger here. And then that's going to pass on a signal. to another messenger. This is going to be another secondary messenger. And then we have another signal, and eventually you have the nucleus. The nucleus has nuclear receptors on its surface. And inside the nucleus, you have DNA. Let's say that this DNA, this gene right here, I'm highlighting in red, let's say that it's turned off. Right now it's turned off. But this signal is going to turn it, whoops, messed that up. Let's try that again. I messed up again. There we go. This signal is going to turn it on. So here's what I'm trying to really convey to everybody is that these signals will cause gene expression. Initially, this gene was turned off. We need it to be turned on. And when it's turned on, it's going to make RNA, right? There's mRNA. That's transcription. And then where does the mRNA go? It leaves the nucleus. It goes to a ribosome. And what is going to take place in the ribosome? Protein synthesis. Here comes the mRNA. That's going to be translation. And then booyah, you got a protein. See all the weird folding I'm doing? Here is the protein. This is what was needed the whole time. That's why that gene was expressed. This protein was needed. And these genes had the ability to make that protein. That's the purpose of signal transduction. And that's the purpose of cell communication. Now that's what we see with a polar message. How about nonpolar? This is actually going to be a whole lot easier. Here's our plasma membrane. It is nonpolar, as Kylie said. What if I had a nonpolar message, Kylie? Could it pass through the plasma membrane?
Yes.
Why?
Because the plasma membrane is also
Nonpolar and nonpolar do mix. So here's a nucleus. Here's the DNA. Let's say right now as it stands, that gene is turned off. This message is going to just do this, everybody. Watch this. Go right in there. Because it doesn't need a transduction pathway. It can go right through. And it's going to turn this gene on. The gene is now activated. It has been expressed, as we like to say. This is going to be gene expression. This is going to lead to mRNA. That whole process is transcription. Then the mRNA is going to leave the nucleus. It's going to go to a ribosome. This is translation. And then that's going to lead to a protein, which is the whole point of doing this, to get that protein. And that protein has a very specific job, and that job needs to be accomplished, which is why the DNA is being told to make it. All right, one last thing I want to be sure I clear up with you all. I'm going to highlight this right here, everything over here in light blue. Everything here is a signal transduction pathway. All right. Nonpolar signals really don't need a pathway because they're nonpolar and they can go right through. All right. Let's talk about when things go right and wrong. Signal transduction pathways influence how the cell responds to its environment. I just thought of a really good example for all you, given that we're in May, summer is about to start. Check this out. A lot of you may be going to the beach or the pool this year and you get a nice looking tan. Tanning is a defense mechanism. Did you know that? We have a pigment in our skin, in our hair, and in our eyes called melanin. And all of you have the same amount of melanocytes. So here's a great example. I just thought of this. Melanocytes produce melanin. This is what gives you your pigment, whether you're dark or light or anywhere in between. It also leads to the cancer melanoma, which is a deadly skin cancer. Well, let's say that you are laying out in the sun and you are exposing your skin to UV radiation. Well, we all have our own tones of skin color, but we all know that melanin, the more melanin you have, the darker you will be. So someone who is tanned has more melanin in their skin than someone who is not tanned. Well, let's say that you're underneath the sun, which is an environmental factor, and that will cause your melanocytes to release more melanin. So let's say here is a melanocyte, and here is a nucleus of the melanocyte. Here is the DNA. Right now, the gene for making more melanin is turned off. Follow me? You go to the pool, you lay down on a pool bed, and you just try to get a tan. The sun is shining down in your body. This is going to be an environmental response, an environmental factor, which is going to cause a signal to be sent to your melanocytes to secrete more melanin. So the gene for secreting melanin is now active. But the actual word that we use is that the gene for creating melanin is going to be expressed. And this will cause your cells to produce more melanin, which will make your skin darker. Darker skin has a protection against UV radiation. People who are very fair-skinned have a higher risk of developing skin cancer than people who are very dark-skinned, because being darker skin is a natural defense towards the sun. It is no coincidence that when you look at countries that have close proximity to the equator, like in Africa, South America, and parts of Asia, natives of those regions have darker skin. And then as you go further away from the equator, like into Europe, they start to have very light skin. That's evolution guided by the environment. So just an example of signal transduction pathways are influenced by responses to the environment. Environment in this case could be like the sun. The sun is going to prompt the more secretion of melanin to defend yourself against that extra UV radiation. All right. Let's see what else. These can alter phenotypes. You know, your skin was fair, now it's dark. That's a phenotype. People have had blonde hair. I had a student last period. He had blonde hair when he was a freshman, and then all of a sudden, his hair became brown. He had the brown gene the whole time. It just had to be expressed, and it wasn't expressed yet. A signal transduction pathway expressed it. Now, I don't know what could have triggered that. Maybe something in the environment. Maybe it was just part of his DNA. He had the methylated gene, and then the gene became demethylated, and it was able to be expressed. The last thing I want to get to when it comes to signal transduction pathways are mutations and substitutions. Remember that receptors are proteins. What are proteins made of? Amino acids. And every protein must have a very specific sequence of amino acids. A very specific sequence of amino acids. If you change that sequence, you may change the function of the protein. So when we talk about substitutions, replacing amino acids may alter protein folding and structure, which could result in a change in function or a lack of function. I told you guys that you all know my name is Kurt, and if there's a substitution to the K in my name to become a C, that doesn't change it. If you spell my name C-U-R-T, but it's still pronounced as Kurt, I'm going to respond to that. But if you were to say Bert, I'm not going to respond to that. Someone says, hey, Bert, come here, Bert. I'm just going to sit there normally. I'm not going to respond to that kind of name. This right here is a silent mutation where it's not going to change anything, but this right here would be a missense mutation where it actually does change. And then we have mutations. Mutations can alter protein folding and functionality because you're altering the amino acid sequence. It alters the codons. And codons code for amino acids. Okay. Obviously, these pathways can be shut down by inhibitors. Signal transduction pathways can be shut down by inhibitors. I want to make that very clear. They can be slowed down or stopped. By inhibitors. And then lastly, I'll let you guys catch up. The last thing I want to get to, maybe a few seconds to get caught up. The last thing I want to get to is feedback mechanisms. There's two types of feedback mechanisms. There's negative feedback. In negative feedback, you want to undo what is occurring. If your blood sugar is too high, lower it. If your blood sugar is too low, elevate it. If your body temperature is too low, elevate it. If your body temperature is too high, lower it. If your blood is too acidic, elevate the pH level. If your blood is too basic, lower the pH level. If your room is messy, clean it up. If you're yelling, stop it. That's negative feedback. Whatever is happening, undo it. And then we have positive feedback. You want to amplify what is occurring. The uterus is contracting to try to push out the baby. Make it contract more so the baby can get out. If your body temperature is rising because you're too cold, rise it some more. If you're making money, make more of it. That's positive feedback. If you're selling product, sell more of it. That's positive feedback. So if something is happening, you want it to happen more, that would be considered a positive feedback loop. All right. So that is it for cell communication. Let's transition into the cell cycle. We have about 20 minutes. The cell cycle is all about the replication of cells. And there's two different types of cell cycle replication processes. This is the only time I'm going to mention meiosis in this lesson. I will talk about meiosis in Unit 5 but this will be the only time. But before I get to meiosis, I would like to talk about mitosis. Mitosis is the replication of diploid somatic cells. If you are having a major brain fart and you do not recall what diploid means, I'll help you out. Diploid means all an organism's chromosomes. For humans, this would be 46. This year's classes have had a lot of trouble remembering that. Humans have 46. An apple tree has 8. A fruit fly has 4. A dog has 78. So a dog's diploid number is 78. A human's diploid number is 46. And then we have somatic cells, which are body cells. On one of the FRQs it was on one of the mock exams, it was talking about a skin cell. It wasn't the point that the cell was a skin cell. It was the fact that a skin cell is a somatic cell. That problem could have said bone cell, brain cell, heart cell, liver cell, lung cell, toe cell. It doesn't matter. It's a body cell. And so therefore it is diploid. The purpose of mitosis is for growth, repair, and replacement. In order for you guys to go from a zygote into a 100 trillion celled adult, you need to grow. That's what childhood and adolescence is all about. You're growing from a child to an adolescent, then from an adolescent to an adult. You're growing, and eventually you'll stop growing, then you just have to replace old cells with new ones. Repair. If you break a bone, you scrape your knee, you tear your skin, you rip a muscle, you rupture a ligament, that needs to heal. That's what mitosis will do. I have a student with a broken arm, and in a nerdy way of me asking, how's your arm feeling? I would say, how's the mitosis going in your arm? That's just, how's your arm healing? That's it. And then replacement. Every three seconds, your body replaces 40,000 cells that just got old and worn out. Cells don't last forever. If you scratch your head, you might see what we call dandruff. That's just skin flakes. They shed naturally. We all shed our skin. And we all replace old cells throughout our body. Bone cells, muscle cells, they all get replaced. I don't know why that mitosis just got messed up there. There we go. And then we have meiosis. Oh, hold on. Before I get to that, I want to make this very clear. In mitosis, you have 2 identical daughter cells. So here's a parent cell, diploid. Here's 2 daughter cells, also diploid. When you have 2 daughter cells, the parent cell is gone. The parent cell is no more. The results are going to be 2 daughter cells. All right, now we can get to meiosis. Meiosis is the production of haploid gametes from a diploid somatic cell. And if you guys really want to impress me, first I'll ask Kylie because she's here in person. Kylie, do you know what type of somatic cell that sperm and egg come from? Okay, she doesn't. So do any of you at home know? Send me a chat on the Zoom text. Do you know what type of somatic cell sperm and egg come from? I'll wait a moment while I'm defining a couple things. If you don't know what haploid means, haploid means half of an organism's chromosome. So for humans, this would be 23. For a dog, this would be 39. And then gametes are sex cells, sperm and egg. Now, keep this in mind. There are 4 unidentical daughter cells. And it looks like this. Here's the diploid cell. It breaks up into 2 cells, and then you get 2 more. 1, 2, 3, 4, and they're all haploid. All right, so let's see if somebody was able to text it to me. One person. All right, Lena got it right. The somatic cell, the diploid somatic cell that all sperm and egg derive from, is called a germline cell. And sometimes there was some confusion, so I want to make sure I'm very clear here. A germline cell is somatic. It will go through meiosis and it will become a sperm or egg. So this diploid cell right here, that's a germline cell. And then these guys here, 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 and here, those are all going to be gametes. Got it? We'll talk more about that in Unit 5, which we will do in the next class. Let's talk about the stages of the cell cycle. Okay, by far, the majority of the cell cycle is interphase. In interphase, the cell is between divisions. It's like intermission at a lightning game. You're in between periods. It's recovering from the previous division. And it's preparing for the next division. This is the majority of the cell cycle. By far, it is the majority. In mammals, it can be up to 90% of the cell cycle. It has 3 main stages. G1, S, and G2. Kylie, do you remember what happens in G1? Organelles replicate. This was a question on mock exam number 2, FRQ 5, C, problem C. Put an X where DNA would be replicated. Some people put an X over G1. That would be incorrect. This is where organelles replicate. Otherwise, cell behaves normally. Also, the cell can double in size because it has to make room for all those organelles. Okay, Kylie, what happens in the S phase? The DNA will replicate. So if you have 1 copy of each of your chromosomes, you're now going to have 2 copies. And then in G2, you do your final preparation for division. Okay, so that's interphase. Now, the rest of it is called M phase. M phase is the actual division of the nucleus and cytoplasm. It is the minority of the cell cycle. M phase has 2 parts. The first part is known as mitosis. Mitosis is going to be nuclear division. You're dividing the nucleus. Mitosis has four basic stages. Number one is prophase. In prophase, you have several things happening. DNA condenses into chromosomes. The nucleus erodes away. This means the DNA is free. And the centrioles move to opposite ends of the cell, or we're going to call those poles, opposite poles of the cell, and assemble the spindle fibers. So prophase actually has the most going on of all the phases of mitosis. So sometimes we break it up into early prophase, and then prophase, and then late prophase, which is sometimes called prometaphase. Yep, I am going to draw it. So here is early prophase, and I'll draw all these. There's our cell. Here's our nucleus. So as you can see, I put some gaps in there. And I want you to notice that the DNA is condensing. Those are now sister chromatids. You can actually see them. That would be early prophase. And then mid prophase, the nucleus is gone and the DNA is free. And then lastly, we have late prophase, which some people like to also call prometaphase. The spindle fibers are starting to align the chromosomes, or these are actually sister chromatids, along the middle of the metaphase, or excuse me, along the middle of the cell, which is known as the metaphase plate. Now, I want to ask you guys, hopefully you can answer at home, how many chromosomes are there? There are 4. There's just 2 copies of the same one. There's chromosome 1, 2, 3, 4. They replicated during S phase. You just have 2 copies of the same chromosome because you're going to divide them into 2 different cells. So this cell has 4 chromosomes. These are known as sister chromatids. Again, this is a chromosome. This is the chromosome just doubled, and that would be a chromatid. All right, everything else should be pretty standard. Metaphase. The chromatids are aligned along the metaphase plate. Why are they lined up along the middle? So they can be split evenly. And I want to be sure you guys understand something here. I want you to look at late prophase. Is that metaphase? It is not. You can tell because they are not perfectly aligned along the metaphase plate yet. And here, they are. They are aligned. All right, next one. Anaphase. The spindle fibers separate chromatids, and they're now chromosomes because they're now separated, and pull them to opposite poles. So here's anaphase. They're being pulled apart to opposite ends. We have less than 5 minutes, so I'm going to pick it up here. Telophase. The nuclei form around chromosomes. Chromosomes decondense. And cleavage furrow forms. Because I'm saying a cleavage furrow, is this a plant cell or is this an animal cell? It is an animal cell, so it looks like this. Here's the new nuclei, and here's the DNA inside. This right here is a cleavage furrow. Animal cells divide from the outside in. If this were a plant cell, everybody, here's how it would look as a plant cell. Here's the new nuclei right there. And as you can see, this little cell plate's forming. That's called the cell plate. Plant cells divide from the inside out. Okay, that's all of mitosis. And the last stage of the M phase is going to be cytokinesis. Cytokinesis is the cell, which we can also refer to as the cytoplasm, divides. You now have two identical daughter cells. All right, I want to draw something really quick, and I have a couple more things, and I'm running low on time, so here we go. This is interphase. We got G1, S, G2. Here is prophase, metaphase, anaphase, telophase, and cytokinesis. This is the M phase. Where are the checkpoints? The checkpoints are here, here, and here. You have one at the end of G1, the end of G2, and the end of metaphase. It's meant to check for damaged DNA. Are these checkpoints foolproof? No, otherwise we would all still be single-celled bacteria swimming in the ocean somewhere. Mutations have to happen, and so these checkpoints are not foolproof. Finally, let's talk about cancer. Cancer is caused by mutations. I would love to spell out the whole definition. I can say it, and you can just write it down when I say it. The uncontrolled growth and division of cells with mutated DNAs. We have P53. This is a tumor suppressor gene. This stops the cell cycle whenever DNA is damaged. If it's mutated, this results in the inability to stop mutations from being passed on. The majority of cancers are caused by mutated P53s. And then last one, I'm going to barely squeeze it in here, are proto-oncogenes. Proto-oncogenes support the cell cycle. It makes the cell cycle go forward. It's like the gas pedal. If it's mutated, then you have oncogenes. And the study of cancer is called oncology because of this. This is uncontrolled cell cycle. This is like your gas pedal in your car being stuck, and it's going to consistently go forward when it shouldn't. So if you have mutations like this can cause cancer to go forward when it should not go forward. All right, sorry I had to rush it there at the end, guys, but I wanted to fit all this in one class period. There's the bell. And that is it for Unit 4. This will be on YouTube. If you're watching this video, you're obviously found it on YouTube. The next lesson will be Unit 5.